Hello, everybody. This is Martin. Me, Keith, and Mark are going to do our presentation on amyotrophic lateral sclerosis and the result that the ER, a little bit of the endoplasmic reticulum, and the Golgi that we learned through this module has on it. Just a little about what's also called Lou Gehrig's disease, who he was, a famous baseball player who was the first actual diagnosis of the disease here in our country. Like I explained, it's kind of the degeneration and the death of the motor nerves. When the motor neurons die, the ability of the brain to initiate control muscle movement is lost. And usually what happens is the patient who has ALS, they usually die from, losing ability to breathe and, swallow and all those involuntary, movements that we have. And they're typically diagnosed, given about 3 to 5 years after their diagnosis. Just a few other statistics or facts here. 90 to 95% are sporadic. 5 to 10% are, genetics. And I wanted to mention this mutation, the SDO1, which is super deoxide demutase. It's pretty important in our future. I want to go over a couple experiments here. Average age of the onset is mid-50s. This was particularly important to me. Military veterans are approximately twice as likely to be diagnosed with the disease as the general public. And then, as you can see here, that there's no no cure, obviously, but there are a couple treatments like Alzheimer's and other Parkinson's that just stop the progression a little bit, but it doesn't cure it. And then we have about 15 new cases a day. So I wanted to show this picture here and kind of talk about the key components. We already learned about the Golgi and how it functions to transport, form its ribbon. It's key to the post-translational modifications of proteins. Here on the left-hand side, I just wanted to show a normal neuron. Dr. Zito mentioned in one of her presentations about the Golgi outpost. So we see here, this is a neuron, which is fairly big, and we see this transport here. And, how we're getting the secretion here. We have the Golgi here, we have the Golgi here. So these would be considered the outposts. But then we have here in an ALS patient, we have this fragmentation here that's preventing secretion, and it's also causing this ALS protein to form. This is kind of the key of ALS. I want to talk about specifically a couple of the key components of the Golgi. Here we see that the ribbon formed here. We see the tubular portions formed. See the cis in the trans side here, the COP1, COP2. So we see here COP1 is very important in the actual transport of the protein from the ER and intra-Golgi and back to the ER. So you have COP1 and COP2. We see here that linked to that is our GM130, which is key to tethering, docking, and fusion machinery. Phosphorylated GM130 leads to dissociation of the actual ribbons or the actual stacks. Also key in microtubule nucleation. And then we have the snares, which would be syntaxin. These are important too. They are main fusion components of the Golgi. So from this cis portion here, which we got the cisterna, we have the syntaxin, which functions to catch that protein as it comes in on the cis side. And then we kind of talked about these grass proteins, Dr. Zito here, how we form actually tubulation between the two stacks. So here in a experiment next, basically they used Western blot antibodies for these. First, the subjects used were SD1 G93A mice and the G85R. So these basically expressed the mutation of that SOD1 that I mentioned. They ultimately proved first that the Golgi fragmentation preceded the onset of ALS in these mice. So that was key to prove first before they started the experiments. Then they used a non-transgenic litter mice and then the wild type SOD1. And what they found was basically they analyzed the Golgi, SDO1 and motor neurons and investigated the COPs, which is those COP1, COP2, the tethers, which is the GM130, and then the snares. What they found was basically they found lower levels of the COP protein. So that would be if you had lower levels, then you would have clusterization of the proteins in the in the cisternae stacks, which ultimately would cause fragmentation of the Golgi. And then they found that the GM130. So here you can see that from the non-transgenic, you see how it loses its here it's not in the SOD1, and it's not. It's faint here in the SOT1 versus non-transgenic, and the GM30 it's fairly equal here. But then they fragmented different fragments here. These P10, P100, and S100 are different fragments. We see the difference here is it's distributed where it's not in the non-transgenic and the wild type. So that's showing that basically you would expect to see it the G130 where the tethering is in the fusion, but they were seeing it in the vesicles and in the cytoplasm, which is not implemented of that it's forming, that it's working correctly. So that was pretty much this experiment. And you'll see in Keith and Mark their experiments too function on some key components that I explained in the slide before. Thank you.
Mark here, working with Keith and Martin. Before I start, I just wanted to make a few notes on ALS that those guys might not have mentioned. First of all, is that there are many mutations that are associated with it. There are some that are much more common, like SOD1, which I think they'll talk about, or chromosome 9, open reading frame 72. And there are others that are much rarer. The other thing I wanted to talk about briefly is the role of the Golgi in ALS. ALS is basically defined by cell death, the death of these neurons, and so they don't know whether the Golgi breaking apart and fragmenting, which is very bad if that happens when it's not supposed to, is what's causing the cell death, or it's sort of happening alongside it or as a result of the cell death. So this particular study was an examination. They did induce an SOD1 mutation, which is a common mutation in ALS, in mice, and later did confirming work in HeLa cells. They mostly used immunohistochemistry and mass spec for this series of experiments, but they did Western blots to confirm that the proteins were being expressed and so on. It was mainly a proteome analysis, so they induced this SOD1 mutation to see what effect this would have on other proteins. And they found, wrote in results, is downregulation of stathmin, this protein, and upregulation of HSP25, HSP27, and peroxyredoxin 6. But it's a little interesting that they saw a downregulation of stathmin because it's a protein that sort of helps break up the Golgi. It's a microtubule destabilizing protein, so you'd think it'd be upregulated. And they do see a lot of it in these fragmenting Golgi, but they saw a downregulation of it a little bit, but they think that might have been a certain form of stathmin. HSP25 and HSP27 are chaperone proteins, they're heat shock proteins, and peroxyredoxin is an antioxidant protein, so the idea was those were being upregulated in response to the damage being done to the cell. And interestingly, they seem to believe that SOD1 was sort of binding with and inhibiting the sort of protective roles of one of those heat shock proteins. So this is examining some of the stuff that happens when the Golgi breaks up. This other paper that I looked at, and I'm sorry, I should say that in the last slide there was no images because I couldn't find one I really liked. For us, there's a lot of images of stains. In this one, they weren't actually looking at an ALS model, but they were looking at what knockdown of optineurin would do in this mouse cell line, mouse neuroblastoma cells. And optineurin is a protein that sometimes sees mutations in ALS, and that is associated with basically making the Golgi work for vesicular transport, for exocytosis. It binds another protein to the Golgi. And so they did a knockdown of it with small interfering RNA and looked at what happened. And they found out that basically knocking down this protein with siRNA caused a vast increase in cell death. You can see it in A in the figure and a little bit in the tissues, but you can see it pretty clearly in B and C, where when you have the OPTN, which is the optineuron, interfering RNA, you're having, very decreased live cell count and increased dead cell count. So interference with this was causing the cells to die. And they believe this was because optineurin suppresses the NFKB pathway. So without it, that pathway is expressed. And there are factors in that pathway that can induce apoptosis. We studied P53 in another class. I'm sure most of you know it, the cancer gene. So NFKB induces P53 expression, or other factors expressed that can induce apoptosis if something bad happens to the cell. And a very neat bit of experimenting that they did is they had the small interfering RNAs that were knocking down optineurin, but they also, once they had that, they transfected a vector expressing wild-type optineurin to see if that would rescue the cells, and it did. And you can see a bit of that in figure D, in D of the figure. Expressing wild-type optineurin in addition to the small interfering RNAs was able to rescue the cells and bring back the survival. And this is relevant because in ALS there's not exactly a knockdown, but there is, in some forms, there's a mutation of optineurin, and so the idea is that could be maybe mean that it's not repressing NFKB properly, and which will lead to the apoptosis of the cells. And as you know, ALS is about neuronal cell death, so this could really be a causative factor in it. Thank you.
Hello everyone, this is Keith, and as part of the group project, I'll be talking about Golgi fragmentation and tubulin binding cofactor E. So a brief introduction to the paper that I'll be talking about. Microtubules are polymers of tubulin, and they've been implicated in maintaining the Golgi organization, as you can see on the right. And just to show how prominent they are in neurons, these are neurons that have been stained with a tubulin-specific antibody. And in the paper, the authors use a progressive motor neuronopathy mouse model of ALS. And interestingly, the axons within the neurons of this mouse die. However, the cell body remains intact, so it enables researchers to actually study things like the Golgi. And in the PMN mice, they contain a mutant tubulin binding cofactor E. The equivalent in humans is TUB4A. And TBCE is essential for the polymerization of tubulin into microtubules. So any defects in this protein is going to be bad for the Golgi. So using fluorescent microscopy, they imaged wild-type Golgi, and they found it was normal, and they also looked at the fragmented Golgi in the PMN mice. They also used electron microscopy. Looking at wild-type mice, you can see the Golgi has the characteristic folds and stacks, whereas in the PMN mice, you see there's an increase in vesiculation and there's just a lack of structure. So based on the evidence for fragmentation, the authors wanted to explore why this might be occurring. So they looked at some vesicle proteins, namely the COPI, and they looked at the beta COP and epsilon COP subunit. They looked at COP2, and they looked at clathrin proteins. And using Western blot, they looked at the relative levels of these proteins in the wild-type and PMN mouse, and they found that there was a reduction in the COP1 proteins, the beta COP and epsilon COP subunits in the PMN mice as compared to wild-type. Using a different cell line, they also looked at TBCE and how that affects beta COP levels. So they used short interfering RNA to knock down TBCE, and they found that in doing so, they also reduced beta COP proteins. So once they saw this, they wanted to see what else might be affecting the increased vesiculation within the cells. So they looked at ARF1, which is a ADP ribosylation factor. It's a small GTP binding protein that recruits COP1 to membranes. So the authors asked if this ARF1 regulates TBCE activity. So they used NSC34 cells to evaluate ARF1 mediated effects on COP1 and TBCE, and they found that ARF1 localizes to the Golgi and increases microtubule formation, as seen here in this confocal imaging. So they overexpressed ARF1 that expressed GFP, and they found, as you can see here in this panel, that beta COP localizes to the Golgi, and there's an increase in microtubule formation in comparison to the normal ARF1 expression. They also used an ARF1 mutant, which is constitutively active, and they found that there was an increased localization of beta COP and increased amount of microtubule formation. So they also looked at the TBCE knockdown and ARF1 overexpression, and they saw that this overexpression rescues decreased beta COP expression that we originally saw when TBCE was knocked down, and it also increased microtubule polymerization. So they used the short interfering TBCE RNA, and they found that when they knocked it down in the wild type, there was this there was the decrease in beta COP here, decrease in tubulin formation. However, in the ARF1 expressed cells, they found that there was a rescue of the beta COP and increased microtubule formation. They also wanted to see if ARF1 expression increased TBCE levels, and using flow cytometry, they found that it did. So to sum up, the loss of TBCE in PMN spinal mononeurons and in TBCE depleted NSC34 cells impairs Golgi microtubules, and TBCE loss results in Golgi fragmentation and decreases in COPI vesicle subunits, as well as increased vesiculation. And importantly, the enhanced expression of ARF1 drives COP1 vesicle formation and recruits TBCE to the Golgi membrane, and at the same time rescues the polymerization of microtubules and prevents Golgi fragmentation. So collectively, all the experiments that we've seen here hint at the possible causes of ALS.